questions. We had one up here, and I just wanted everybody to hear it because when somebody has a question, a lot of people have the same questions, and some people just won't ask it. So, go ahead.
So my question was, when you're kind of talking about the autologous versus allogeneic umbilical stem cells, to say one is better than the other, it's all a in vitro kind of
Correct.
explanation right now, being telomere length or doubling. Is there any studies that actually show a in vivo process of showing the two, like a side-by-side comparison?
No, there's none out there that nobody's done that because they're not. These type of studies in humans cost a lot of money, and it's difficult to see. So everything is normally done in vitro, which even costs a lot of money. But what we do know is that the younger cell, and it makes sense, the younger cell secretes a lot more factors. It works much better in animal models, and this is what we know so far. Somebody eventually will probably step up and pay some money to look at differences in one type versus the other. But for instance, the only comparison study that I know of that's been published is in looking at autologous MSCs versus autologous chondrocytes in joint. And what they found was that they were both equivalent. The MSCs were a lot more potent because you could obtain the MSCs from any age patient that was there, and they worked much better in that aspect, meaning the aged MSCs were better than aged chondrocytes. Secondarily, when you're talking about using cartilage, you're now talking about extracting cartilage from the patient, having to grow up. So you're talking about two different procedures in the same exact joint. So that's problematic right there. So in essence, what they basically came to the conclusion was that the MSCs were better than autologous cartilage. Those are the only studies that I've seen as far as comparison goes in humans. Yeah. Any other question? Yes.
Just about telomeres, it's a little bit on the side, but you recommending any supplement to protect the telomeres?
The only thing that's proven is caloric restriction, as far as I can tell. Astragalus, as you've heard of, it's one of the things that works well in, for instance, some of the TA65 or whatever. But if you read the publication well, the publication discussed only about 30% of the patients' telomeres improved. Some of the patients actually got worse, and some of the patients did nothing. So it just depends. I think it depends on the individual. It's a hard thing. I think, diet restriction, if you think about it, if you cut calories or if you stop eating, your body goes into starvation. It goes into, I can't divide, I can't do anything, I got to stop dividing. The more you consume, your liver's in overtime, everything else is in overtime, actually duplicating cells. The more cells duplicate, the older you get. If you look at, for instance, IGF-1 or the use of HGH, the HGH individuals have phenomenal results. But at the same time, if you look at all the animal work on using these type of things, you put yourself at risk of many different things because eventually this type of division, because IGF causes cell division and a lot of cell division, because that's what transpires when you're younger, is how you feel better. And it's problematic because you have to look at, in mouse models, it causes early aging, eventually it catches up, these type of things. However, this is only one mechanism. Remember, your body has various different lines and various different mechanisms of aging. For instance, stem cells or senescence are not just one. Mitochondrial health is actually another one, wiping out these old cells to force your body to regenerate, maintaining health and overall. It's like, for instance, the individual that I get regularly. So you'll get patients, and you guys are going to see this, that are going to come to you and, hey, I have COPD. I'm a chronic smoker for 50 years or whatnot, whatever it is. And you tell them, okay, fine, this can help you. What are you doing now to help yourself, or what are your plans? And then you have the patient that tells you, well, did you quit smoking? I did not quit smoking. Do you plan on quit smoking? I don't plan on quit smoking. So that's a simple thing as to don't waste my time, because you're wasting your own time and your own money, because if you don't help me, I can't help you. And this is very, very common throughout the board with this, because this is not just a one thing. This is a change in your life of taking specific steps. For instance, I'm a strong proponent of prepping the body before you actually do this. And this is really, really important. Because if you put cells, and I've got a ton of data on this stuff in vitro. If you put a cell in a harsh environment, it goes like this. If you put a cell in an ideal environment, it lasts longer. And when it lasts longer, because remember what we said, these cells try to survive. They're in a condition that they're not used to. They try to survive, even in blood flow, even in the joint especially. The joint is really harsh. It's synovial fluid. It has specific proteins. It's not set up to survive. It's set up to try its best to survive. But if there's a massive inflammatory response in that joint already and you throw it in there, it's going to get wiped out relatively quickly, what you're doing. It's going to do its job, but it's going to get wiped out. But if you prepped the body beforehand to help suppress the inflammatory response, for an individual to eat the right way, for an individual to take the right courses before they're actually doing this, you've now set yourself up and you've now set the patient up for success. And it's important. It's not just a one and that's it. This is not, this is not a Band-Aid, is what it basically boils down to. Yes?
First of all, I enjoyed your lecture. It's very, very informative.
Thank you.
Second of all, I just wonder, what is your opinion regarding the initial Steam Black Research Institute saying that the bone marrow and the adipose stem cells has less quality and quantity as compared to his umbilical cord stem cells? That still holds as they believe, you believe? Is it still true that
This is what we just discussed right now is yes, I mean, as you age and depending on the condition, the individuals that are coming to you are individuals that are going to be most likely older, problematic. I mean, there's a ton of stuff going on. You've been exposed on a daily basis to, for your whole life to DNA mutations, whether you go out in the sun or whatnot. Your immune system has to be optimal. It has to work the right way to actually combat all these type of things, and it becomes problematic. I mean, the lady that died like 2 years ago in France, she died of stem cell exhaustion. Eventually your cells multiply, but she was like 100 something. She was the oldest lady, I think 120, something like that. I'm not sure the exact number, but she died of this is what when they looked at her, when they studied, this is what they had found, and this is. I mean, she lived 120 years. That's a long time. But it's something. It just depends on what you're eating, what you're. My personal opinion is there's nothing more important than what you consume and what you put in your body. There really isn't, because you can work out day in, day night, 8 days a week. And if you're not eating right, you can have a fat layer over it. You're not going to look good. You're just going to, it's all about consumption and nutrients. Nutrition is the most important thing. Yes?
Thank you. Thank you.
You're welcome.
You had mentioned that stem cells were used in some of the autoimmune conditions like MS, and it caused modulation of the immune system. Does that mean that the immune system got better, or decreased the immune system in response to the MS, or?
So it basically means that you have, it regulates the immune system. The Th2, which is a pro-inflammatory response, comes up, and the Th2 anti-inflammatory response, comes up. The pro-inflammatory response actually goes down. The vast majority of these patients run on pure pro-inflammatory because it's an autoimmune disease that's consistently on attack mode, or prepared for attack. The most important aspect is that regulatory T cell is increased, and it doesn't allow the immune system to go into a pro-inflammatory mode, because it regulates it and it says, hold on a second, I don't need you to attack, I don't need you to, to go into attacking this tissue and killing this tissue off or become reactive. So that's the way it works, basically. It's basically what we're looking at is changing an immune system that's in disarray to making this immune system homeostatic. And that's it. Put it in homeostasis. Yes?
You touched upon the prepping patient before the stem cell therapy. Is there any specific guidelines or any kind of diet therapy that you start that maybe a couple weeks prior to that treatment?
I mean that you can. I'm more of a intermittent fasting type of individual, eating right, keeping carbs down to bare minimum. But carbs are necessary too, though. I mean, if you think of all this anti-carbs, think of the old gladiator and warrior days that those guys were shredded, ripped. What did those guys eat all day? Grains, bread. But the problem is that the consumption was kept at bare minimum. The consumption, the amount of food. I mean, here you go to a restaurant, and what do you get? Go to Europe and go to a restaurant, and you get a plate of food that's about this big. You know, come here to any restaurant, you get a plate of food that's like that big. I mean, it's just the amount of calories you're consuming and taking in. It's destructive. I mean, it's destructive to everything.
As far as the supporting and the supporting therapy after the treatment, the stem cell treatment, any kind of supplementation?
Same thing, eating correctly, don't doing anything. And I'm real big on testing prior to looking at food allergies. Before you do this, see what's going on, and then removing those allergens, and then and then eventually bringing them back on to see which ones are problematic, this type of thing. But that takes a long time. Sometimes these people are really sick or they're problematic, whatnot, and they don't really have time for that. Supplementation, as far as supplementation goes, probably 75%, including myself here, require vitamin D on a daily basis. We are almost all deficient in vitamin D, one of the key things. Vitamin C is really important. Omegas, I think everybody should be on N-acetylcysteine. I mean, I'm not telling you what to do, but this is my personal opinion, or not telling anybody what to do, because you want to keep that glutathione and keep the oxidative stress under control. That's the #1 problematic issue a lot of times in the body, because it's not released as we age in the right amount. This type of stuff is really, really important. A B complex is important, but B has to be watched. I mean, it just depends. Everybody's different. Any other questions? Yes.
So we spoke about the inflammatory response and how this helps modulate it or put it in homeostasis. Medical school 101 kind of education is that inflammation is not always a bad thing. It's helping the body to heal. So are we assuming that there is an over-response in these individuals? That's why they're presenting the way they are, and this helps control that?
In autoimmune disease, the problem is there's a consistent response to an antigen that is basically present that it's recognizing as foreign. So you have sort of the immune system on pro-inflammatory response mode consistently. Yes, a pro-inflammatory response is absolutely needed, but if the pro-inflammatory response is not working the correct way, then you're not going to get healing. You're not going to get the correct healing. So if this pro-inflammatory response, so if you look at the charts and you look at the data on, for instance, PRP is really interesting. If this pro-inflammatory response is a slow and low modulated inflammatory response that lasts a really long time, that anti-inflammatory response that's going to come in and do the healing is going to be very, very similar and not effective. So in the case of PRP, you cause a massive pro-inflammatory response. And from that massive pro-inflammatory response, you get a massive anti-inflammatory response, which is the healing response to it. That's why that works. And it's a very similar concept. When you fall when you were a kid, you get that massive pro-inflammatory response that's relatively quick. Kids cry, they're in pain. Remember when you fell, you cried, you're in pain. How many times do you cry as adult, we fall? How many times have you cried? It's been painful when you fall, but how many times have you cried as an adult when you've fallen down? Because the pain, the agony, it's not as bad as when you were a kid. It's because that response is so immediate, so quick, and then that secondary response is so immediate, so quick, and that's why the proper healing occurs. Any other questions before we go? Yes?
Has anybody looked at non-steroidals and other anti-inflammatories after stem cell treatment to see how it affects the treatment negatively or positively?
Oh, there's a lot of conflicting reports on that. NSAIDs are not good because you want to keep that response, that immune response, okay. Steroids are used commonly, though, believe it or not, because, well, the mechanism is somewhat different because they don't work directly with prostaglandins, and it's a little bit different. The mechanism is a little bit different. Steroids are used, for instance, in bone marrow. The standard of care is, these patients that get allogeneic transplant, because you have to, you give them an H1 and you give them an H2, and then you give them dexamethasone, which is common. And it's because you avoid, it's a prophylactic to avoid any form of reaction or anything like that. And engraftment still occurs, and the cells work perfectly fine. In instance, for instance, when we do this stuff offshore for IVs, we follow the same protocol because this is a protocol that's been established for years and years, and we've had a lot of great efficacy in this.
People not to use anti-inflammatories after
NSAIDs, correct, yes, yep, yeah. We do. All right, let's move forward. The next thing we're going to talk about is actually a new, relative, it's not new, it's an old field, but it's something that you're hearing a lot and a lot more about. And this is now, considered one of the future regenerative medicines, which is exosomes. Going back to this name, remember I told you you're going to hear some things repetitively here. We're going to go back to this cartoon right here and this little picture right here, that the cell is the smallest unit of life, and it requires communication, and this communication is secretory vesicles. Exosomes fall under these secretory vesicles, and they are what help, not only thing that helps, but they are one of the most important aspects that make changes, physical changes from one cell to another, physical changes in production of extracellular matrix, of almost anything that is actually done. The cell not only secretes what's called first an endosome, but it also secretes different proteins and these type of things on its own that are being produced, remember, within the cell. All this stuff is being produced within the cell, and then it's eventually secreted off and blebbed off. These extracellular matrices actually started with, you hear them from mesenchymal stem cells, but cancer cells have exosomes. Any cell type in the body basically produces an exosome. It's from cells. They produce this, and it was actually, the early work was actually done from, believe it or not, from red blood cells and from platelets that produce it. And these were considered, what are these discards? We didn't know what it was. Are these just cellular debris? What's transpiring? They didn't know what it actually was. Eventually they actually, these are called exosomes microvesicles, and they actually found what they were, because there's various different types of vesicles that we actually secrete, and they vary quite a bit in range. These vesicles can transfer proteins, messenger RNA, microRNAs into other cells, and this is done sort of like a mechanism of action of blebbing. In the cell, it forms this vesicle that is very similar to a cell. It's a bilayer, lipid bilayer, and then eventually this thing migrates off of the cell and it blebs off, and it then goes to a partner cell, and it latches onto that partner cell, and the information's engulfed inside of that partner cell. These things range in size because we're talking about extracellular vesicles or exosomes. Exosomes are relatively small. They're 40 to 100 nanometers in size. So what we're discussing is about a, what is that, about a 10 to the 9, 10, I'm sorry, 10 to the 6. It's about a thousandth, less than a thousandth of the size of a cell, depending on the cell that we're actually using. So they're relatively small. Microvesicles are even smaller, and these apoptotic bodies are even smaller. These are just really, really small little bleb vesicles that are actually released from cells. These are affected by various different things that transpire on a normal basis in your body, what's being produced from these. So whether it's age, gender, disease, fasting state, medication, etc. Cells respond to the environment. This is a take-home message for these things. They secrete what's in that environment, what they need to in that environment. If the cell's in an ideal environment and it's functioning phenomenal, it releases what's maintained for homeostasis. It's going to continue to secrete that until some form of a disarray or some form of disease or something is there that makes it secrete something that it doesn't, that it shouldn't. The Nobel Prize in 2013 was actually won on actually looking at a regulation of vesicle traffic for this. So it's actually a very, very interesting field and a growing field. Here it is. In this cartoon right here is sort of what I had described to you earlier, when these things are first endosomes and then they're secreted out as exosomes. For those that tell you that, this is better than a cell, don't tell me that, because the cell is what produced this. And inside the cell, it's in there as an endosome. So if I take a cell and I rupture a cell, an endosome is in there that's going to be delivered out as an exosome. So please don't tell me that, because it really doesn't make that much sense. Especially, if you think of it functionally, and if you talk to cell biologists and you talk to, any individual that'll tell you about this. Yes, it works, and the concept is phenomenal. It does a change, which is what it's supposed to do. But working with cells for me is a lot better source because you have something that's constitutively expressing this stuff. Until this stuff dies off, it has no choice. Until cells die off when you put them somewhere, they have no choice to help feed my buddy that's sitting right next to me, that's another cell, has to feed off of me. And I got to spew this stuff up to help him out. Otherwise, he won't survive. And he's got to do the same thing. He's in the same autopilot mode of spewing this out until eventually he dies. And everything that he secreted out is gone. This can take anywhere from, let's say, 3 days, a week, 14 days. We have evidence in animal models up to 14 days that the cells survive. So they release anything from, protein, lipids. They actually have a lot of lipids in them. And lipids, of course, are good. Everybody, cholesterol is bad. Cholesterol is not bad. Just controlled cholesterol is bad and done the right and problematic. They release quite a bit. They serve as vehicles to transfer bioactive cargoes. There's been over 700 different identified in bone marrow mesenchymal stem cells alone, and there's 420 that are similar in other cell types that are out there. Over 200 different transcripts of mRNA are found in MSC's exosomes. And these miRNAs, which are really, really small fragments of coding, non-coding actually, small regulatory, these actually get delivered to specific target cells that can make a change. Lipids, they contain quite an abundance of lipids because remember, they're wrapped in a lipid bilayer, just like a cell. It's just consider a smaller cell type without all the organelles that are actually in it. And then a lot of these lipids serve an importance in different things. Believe it or not, this, which we'll discuss a little bit more, one of the biggest fields that is actually targeted right now for this is actually for neurological disorders, because it makes sense. I mean, look at all the lipids right here, and it makes complete sense. These secrete an abundance of these different things. Heat shock proteins, everybody remember from school, or did they ever teach you in medical school what heat shock proteins serve? Remember, they're very, very protectant against oxidative stress and many different things. These tetraspanins are actually how we identify that we actually have an exosome. And once again, you keep seeing these CD. A lot of people, what the hell is a CD? It's cluster differentiation. It's a marker on the outside of a cell. Believe it or not, when I went to school, I think we were at about 160. We are now at about 400 of them that have been identified in cells around there. And it's a growing field because people are finding more and more new proteins. And cells change, configure depending on disease, depending on what they've been exposed to. That'll express different ones that are actually found. An abundance of lipids that are secreted from these, different nucleic acids that deal with coding and actually non-coding. So you're talking about something that's a bioactive. Okay, these microvesicles derive from, for instance, endothelial cells. What we discussed is a really, really important cell type in your body, probably one of the most important cell types in your body. Activate angiogenesis through the transfer of proangiogenic molecules. Remember, just want you to see these consistent vascular endothelial growth factor. It's important, increases vasculature. Basic fibroblast growth factor. It's important to strong mitogen, keeps youth going, right? Platelet-derived growth factor, very, very similar vascular endothelial growth factor. It's actually known to increase vasculature. Leptins, acidic fibroblast growth factor is another of the fibroblast growth factor that has a specific mechanism of growth and proliferation. TNF alphas, TGF betas, and others are secreted, but actually to work with suppression of these, along with proteases, matrix metalloproteinases, the good kind, because there are good kinds and there are bad kinds. You don't want matrix metalloproteinases in your body if they are specific types like MMP13 that can be detrimental. MMP9 and MMP2 are actually good ones, and so on. They've been shown to, they can actually produce more, believe it or not, more membrane parts, which are fats, and they become activated based on, for instance, monocytes that can help induce apoptosis in a bad situation, or maybe even suppress apoptosis by targeting caspase. Caspase is a family of basically activators of cell death and apoptosis. There's anywhere from caspase 1 to, maybe I forgot some of my biology, but I think we're down to like caspase 14, depending on if it's intrinsic within the cell or signal from outside the cell, which is mediated by this cluster differentiation CD40 that causes activation of apoptosis on a regular basis. Probably the strongest one that does that. They're rich in annexins. Annexins are really, really important in cell-cell connection, and then also in mediating apoptosis. Tetraspanins are these markers that we actually look at, increasing and dealing with heat shock proteins. They expose low amounts of phosphatidylserine and specific types of cell type specific proteins that are outside, on the outside of the cell. They deal with tumor susceptibility gene 1 also. Clathrin, which actually deals with antimicrobial, are also frequent in some of these things also. This is in essence how these things work, and this is the case in just, we're just discussing, but just think of any cell type in your body of how they do this. And the stem cell in this case is a stem cell can secrete this thing, and it can take a injured cell, and then it connects, and it's taken in, and it makes a change in that injured cell. In that case, it directly stimulates in this. In this one right here, this case, it's looking at transferring genetic information to eventually, if this cell needs to divide off, it'll divide off and it'll function the correct way. It can directly deliver proteins to these injured cells, meaning this is a protein information packet, and it delivers it because this cell needs that protein to survive and it needs it for work. And then it can transfer receptors. Receptors are on the outside of the cell, and they could also be actually found on inside, and it can be ready for formation. And the purpose of it is because you have proteins that might be floating around that now need some form of activation. The protein is not activated without the receptor. It needs to bind. Remember, everything works on a complementary sequence. You have a receptor and a protein, or a ligand and a receptor. Lock and key system, what we just described. It's the only way things are activated, the way things work in our body. How do you get these things? Normally, you can actually obtain them from ultracentrifugation, which is something that runs about 100,000 Gs or faster. And these things will migrate all the way down because you're spinning it so rapidly, everything migrates down. And then you can identify them by what's called electron microscopy. It's a very, very expensive piece of equipment where you can actually see them because remember, these things are so small. We're talking about a piece of equipment that runs about $1 million to look at them. Atomic force microscopy is also very, very expensive. Non-particular tracking analysis is actually something, tracking analysis, something that's relatively newer now. These machines, all of these equipments to look at things that are so small are really expensive. We're talking about $500,000 on up to identify them. Simpler methods we look at them by using what we said, flow cytometry, where we look at the specific marker like what we saw earlier, CD9, CD63. We do Western blotting on them. Cytometric analysis, what I just discussed, Western blotting on them. And then we look at ELISA. ELISA is basically to identify, all you guys know what ELISA is, but it's basically to identify specific proteins that we want to look at that may be secreted from these exosomes. Here's a EM structure. This is very, very detailed. Just think of this thing being, it's impossible to look at. I have a scope that goes, 100x scope times 10, so it goes 1,000x. There's no way I'd be able to see anything like this, and that scope runs about $100,000. There's no way I'd be able to see anything like this. So this is really, really tiny, and you can actually see the blebbing of it, or being in contact with another cell in here. And then you look at the analyzed protein and RNA. And the interesting thing about this article, and then do it by Western blotting, looking at specific things, looking at what they want to look at. The interesting thing about this article is what they determine in this article is that you really don't need that much of this for an effect. So this runs in microgram amounts to have an effect. You'll probably hear out there about products that are available out there that talk to you about milligram amounts. I find it hard to believe. These are products that are most likely supernatant in nature, meaning there's a difference. The vast majority out there, like we talk about stem cells, and everything in there is a bunch of other cells, so it's a stem cell-based product. Same as some of these exosome-based products that are out there, including one that I actually have. It's basically, you put the cells in a condition for them to secrete, and you recycle what you secreted. And in there going to be exosomes in microgram amounts, but there's also a bunch of other beneficial stuff. Remember, cells secrete extracellular matrix, hyaluronic acid, etc., which we'll talk about in just a second, that these things secrete. Here's a really, really interesting article that's being, and there's more and more articles popping up like this. Exosomes from umbilical cord blood accelerate cutaneous wound healing through, this is a specific mechanism of what's being secreted from the exosome. It's a specific miRNA-21-3P. And this actually promotes angiogenesis and it increases fibroblast function. This tells you, if you think of it logically, this probably serves as not only a healing effect on a wound, but secondarily cosmetics, right? So it should work in cosmetic. Products that we've tested and products that we've actually looked at, and then we look at, and one that we actually produce, like I said, it's what the cells secrete, has collagen type 1,3, hyaluronic acid, and chondroitin sulfate. Stem cell factors that are actually in there, nerve growth factor, basic fiber, FGF that we discussed. Stem cell factor, which actually is also called c-KIT, this activates your own stem cells in your body. Vascular endothelial growth factor, VEGF, and many more that you'll see in a little bit. We look at different methods of producing this stuff. How do we produce more versus less in a simplified method? And then, sorry that this is cut off, but there should have been another line underneath here. And it just basically describes 3 different methods, and this is one of the methods that we found, that if we take and we do a specific method of it, we actually get a 10-fold increase in collagen type 1 and 3, because remember, collagen is the most abundant protein in your body. This is actually some work that we did with Cleveland Clinic, and we looked at B stands for bone marrow, U stands for umbilical cord. We looked at methods of basically causing the optimal amount of hyaluronic acid and chondroitin sulfate for orthopedics, or actually for another thing, for skin. And we actually found that this particular media formulation was better. This is the media, what's being secreted. This is what's inside the cell. So remember what we discussed a little while ago? People tell you that, the cell, blah, 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 this is that. Look at the difference in how much of this stuff is being manufactured inside the cell versus what's being excreted out. It's a big difference. And even in the optimal formulation that we found, that was this one right here, this is basically running different formulations of food to see what is ideal, what's optimal to get this particular product to secrete out this for use. And the nice thing about this is that we did some work with them, is that it's not bad hyaluronic acid, because not all hyaluronic acid is actually equal. So low molecular weight hyaluronic acid is actually not really good for you, not beneficial at all. You want the stuff to be over 5,000 kilodaltons. Synvisc, the commercially available product, runs in the range of, if I'm not mistaken, about 8,000 or 9,000 kilodaltons. But this is a modified hyaluronic acid that's been produced with modifications to it to increase the molecular weight of this. The stuff that we produce here is greater than 5,000 kilodalton. Growth factors that we've tested in those same exact different formulations, we came to the conclusion, a specific formulation that we found that gives us the optimal amount of growth factors. And we found this to be better in umbilical cord versus the bone marrow that we're using. Here's a list of the different things that we actually looked at. In general, different things that we looked at that are being secreted by these different factors. Some of these are actually pro-inflammatory in nature. And the take-home message that I put this up here for is, I see companies that post these things with probably about 50 or 100 of these different factors. And people say, wow, this stuff has all this in there, that's phenomenal. But have you thought about what some of this stuff does? So this is a pro-inflammatory marker. So is it going to be problematic? There's several anti-inflammatory markers here that are actually good. But this is a pro-inflammatory marker too. But maybe it serves a purpose, and we don't really know. This stuff is secreted. I'm just giving you an idea of this, because I have seen entities that post 50 of these, and somebody will come up to me and say, hey, can you produce a product that produces all this stuff? And I look at it and I said, why? Some of this stuff is bad if I look at it. I don't want to do something like that. I'm trying to focus on a particular thing. For us, the importance of extracellular matrix, cells don't function, proteins don't function without that around. You need to have extracellular matrix. Like I said, there is nothing more abundant in your body than collagen. These serve based on, for instance, the data that I just showed you. This is internal data from some of the work that we've done. It serves well for, I mean, if you think about it logically, what we've seen, orthopedics, wound care, cosmetics, and hair restoration. You know, if you look at it, and it does based on some data that we have that we're running studies on, it does serve really, really well to do this kind of stuff. This is something that just came out. This is a fresh publication that came out, I think, a week ago, if I'm not mistaken. Exosomes for aging. What they found here was they looked at, and this is once again in vitro. This is, you know, in a petri dish. They looked at two different types of exosomes. Exosomes from a mesenchymal stem cell, and they looked at exosomes from inducible pluripotent stem cell. Does everybody know what an inducible pluripotent stem cell is? How it's produced? Should I go over it? Let's go over it real quick. So it's basically, there's various ways to do it, but I take the old way, the original way of doing it is I take a skin plug from any one of you, and I add 3 or 4 very young and embryonic genes to it in culture with a virus. Now you can actually do it with protein expressions because we stay away from using viral infections with these things. And I add this in culture, and I go back and I take your old cells and I reprogram them into an embryonic state. So I'm reprogramming the cell to a very, very naive state. I can now take your cell that I've actually programmed and produce any cell I want in the body. It's an embryonic cell. I can produce any cell in the body now. And this is what's an inducible pluripotent stem cell. This is possibly the future. The only issue that they're finding with many of these is very, very similar to, it's good to you know, you can recapitulate models of disease with this. Because if I obtain it from an ALS patient, think about this, and very, very similar to, remember Dolly? Dolly was cloned. Dolly died like that. It's because everybody carries inherent methylation patterns. Everybody carries these genetic issues, and they'll age rapidly. Very, very similar to these type of models. But they're used disease models. They work really, really well to study. And possibly in some future, they'll be able to solve these type of issues with these cell types. But in this study right here, they took MSCs from adult. They took and they produced exosomes from it, and they produced exosomes from iPSCs, which are much younger. They found, if I'm not mistaken, it was like 100-fold or 16-fold, I'm sorry, more of these exosomes in the iPSCs than they did in the MSCs. But the problem, the funniest thing about it is that the ones that were from the inducible pluripotent stem cells had greater antioxidant effects than the regular MSCs did, but the results are relatively similar. There wasn't that much of a they saw some decrease in the formation of oxidative stress of it. When we talk about senescent cells, they basically took skin fibroblasts, they plated on a plate, they aged them naturally, they made sure that they're senescent, they can no longer divide, and then add these things to them and see, because senescent cells secrete what's called senescent-associated factors that are detrimental. We now know that this stuff causes cancer, heart disease, dementia, etc. That's why aging sucks, guys. It really does. But anyway, they found a way sort of using this to mitigate the oxidative stress that was actually released by these cells using these things. So it's pretty interesting. And then the other thing else, like the neurological disorders, I have a lot of colleagues that are actually using this in neurological disorders. And if you look at what's secreted by it, it makes sense. You know, and it's something simple that's relatively simply produced without cells that you can probably have, you know, this is an effect, and then, you know, you have something that you've produced and it can cause an effect, a quick effect, because that's the way cells work, versus just using cells. And it would be much easier, I believe it would be much easier to get this stuff through the FDA and through regulations of this stuff. Young versus older cells, different types of stem cells. We know this for fact, based on what the study we've done. We've looked at, like I said, remember the bone marrow I showed you versus, those are from a 40-year-old bone marrow versus a younger one. The production is different, doesn't necessarily mean it's bad, because the function of this stuff is for healing anyway. The growth conditions are really, really important, because this is what's responsible for what's secreted. When they're in an ideal, happy situation, they secrete happy stuff, and they secrete stuff to maintain homeostasis, healing, repair. When they're in a stress situation, they're like on overload, and they got to find a way to fix things. One of the other things that we don't know, you know, what these things release. How do we control what they release? we have somewhat of an idea by playing with culture conditions, but it's difficult. We still need to work on these type of things for exosomes. And for in general, what I call actually is a supernatant. I don't even call the products I work with and the products that are out there right now are supernatant derived. They're not purified exosome products. Okay, so this next one we're going to discuss is adhering to quality standards of regenerative medicine and for regenerative medicine. This is probably the most important, this is the most important talk that I'm actually giving here, I think personally, because this aspect right here is not only for your protection, but it's for your patient's protection. For instance, when a product, and I'm going to go through basically products that are produced, products that are given, whether it be autologous from that patient or whether it be an allogeneic product. Anything that is done and anything that is produced has to be with the correct informed consents. There is nothing more important than that, guys, please. Does your informed consent cover that this is an experimental procedure? Does your informed consent cover the risk, simple risk of when you inject a site, did you put in your informed consent that even when you inject anything in, there's a risk of infection. Is that in your informed consent? Simple things like this is things that we take for granted, and you're not covering yourself if the informed consent is not correct. Is a donor pre-screened, whether it's autologous or whether it's allogeneic, either way? Is the donor pre-screened? Does the patient have a predisposition of cancer? Did they have cancer? You're injecting a joint. Did the patient have a bacterial infection previously to you injecting the joint? Because I'm telling you right now, if they did, you're looking for trouble if you inject that joint because you're going to activate that bacterial infection that's dormant. It's simple things like this, questions that you need to ask your patients, and you need to make sure they're crystal clear. There's nothing more that, I'm not a practitioner, but I've dealt directly with patients with colleagues of mine that are a practitioner, and I really, really hate when you ask the question, what have you taken today, or what, and then all of a sudden, when you're into a procedure, or they're feeling a little weird or whatever, or something's a little going south, they, oh, I forgot to tell you I did X, Y, and Z. There's nothing worse than that. And it's just, you know, you got to make sure that you go through all of these questions and all of these concerns beforehand. You know, in this case, when we talk about testing allogeneic product, the donor has to be very vigorously pre-screened. Meaning, is there a predisposition of genetics? Is there a history of genetic issues, cancers, HIVs, whatever, these type of things with the patient. Testing standards according to the state of California are these that I have listed here: HIV 1-2, HLV 1-2, Chagas, West Nile, syphilis, and CMV, Hep B, C. I would tell you when using anything autologous, it's important that you look at Epstein-Barr virus. I have this hypothesis that Epstein-Barr virus is the worst thing that has ever been delivered into this planet. And it's worse. I got a simple question for all you guys, and people get this wrong. Does everybody know the difference between a virus and a bacteria? What's the major difference between a virus and a bacteria? It's a hard question.
Technically survive without a host, because it has everything it needs to survive
Huh?
on its own.
Bacteria can divide.
Well, no. So that's exactly it right there. So the bacteria does not. The virus requires a host, and the bacteria does not require a host. So that's it, basically.
The intracellular obligate parasites like the chlamydia, which are the Q forms of the old days.
Yeah, those. I mean, they require a host too. They require a host, which is similar. But viruses require a host.
I think it's a bacteria.
Yeah, a virus does nothing. Viruses are horrible. I call viruses aliens, actually. They look like aliens when you look at them underneath a scope. So yeah, but they're absolutely atrocious because they're opportunistic. They do what they can. They take total. I mean, yet we live in harmony with some of them that are actually not bad for you, but the vast majority of them are actually really, really bad, and they're opportunistic. I mean, you're stressed, and all of a sudden, what is this horrible thing that comes out? Not chickenpox, the other one, shingles. anything like this. Your immune system's not functioning right, and it's sort of weird how we don't know why some individuals react well to Epstein-Barr, which a lot of us are infected with Epstein-Barr, and some individuals, the vast majority of patients that have cancer, if not all of them, based on my communication with many practitioners, have Epstein-Barr virus and have very high viral titers of such. They're problematic. They're very problematic. Anyway, before any product is produced or before anything, when we talk about allogeneic, this product has to be quarantined, meaning a product is produced and a product is quarantined and all the testing has to be done, and the testing has to be able to be, confirm that everything is clean on it, that there's no problems with it. All of these products should be processed in the correct environment. Allogeneic products, off-the-shelf products, have to be processed in the correct environment, a regulated environment that's ISO 7 or better in this case, a clean room that's monitored for airflow and filtration, weekly cleaning and disinfection by a dedicated cleaning crew that knows what they're doing, and extensive training for going into these rooms and out of these rooms. This particular thing right here, you will see, you'll go somewhere, and they'll tell you, oh, my room is in ISO 7, ISO 5, whatever it may be. I think it's hilarious when people say that I have an ISO 5 clean room, and I'll see somebody going in there without full gear on. I'm like, that's impossible. This room, this right here certificate just means that the room has been certified and built as such. It's now up to the company to maintain that certification. What that requires is every single time product, you see somebody in there, every single time somebody's in there and producing a product, there's also going to be a regulatory or somebody else in there with these little tiny wonderful agar plates and with a particulate read counter in there, making sure that it's maintained according to this little certificate right here. Because if you don't have that, then you don't know. So these are questions that you can ask to the regulatory staff that are producing these type of products, saying, do you have the maintenance of that particular room, showing me that that room is clean when you produce that product. Although testing on the product is done, but that's the only way that, for instance, when the FDA comes and inspects us, it's the first thing they ask for. Show me that you're doing, how much products you're producing, what are you producing, show me that you're running a regular system in there, that you're maintaining those standards, because that's the most important aspect of that room. Processing in clinical grade, not research grade equipment. You're using the correct equipment that's been calibrated on a regular basis, showing you that it's functioning the correct way. These hoods are ISO 5 rooms. The room is ISO 7, for instance, where we do it. And this is a biosafety cabinet, the correct type of biosafety cabinet. There's so many different types. When you talk about doing cell work, you work with these things that are called laminar flow hoods. The air flows down along the bottom and then out into a vented system that's actually right here. The concept is to keep anything in the air. As soon as anything's in the air, if there's any form of bacteria, it spreads it down and shoots it out and shoots it out, down. When you normally run plate reads, and we run plate reads inside of here, you will find nothing, or bare almost next to nothing inside of here. In the oxide room, you find CFUs and you find these things. But every single step of what you're doing has to be optimized. It's up to you as practitioners, whatever you decide to buy, whatever you decide to use, to make sure that this type of quality assurance and quality control is maintained. You got to ask those questions. The processing of it is, are the individuals doing what's called a USP 71, meaning they're outsourcing it. The standard is you outsource it to a CLIA certified lab, and you get records that the stuff is clean. That means the product has to be quarantined, because that takes up to 2 to 3 weeks before the product is released. And even when the product is quarantined, the product is maintained somewhere absolutely different than when it's maintained when the final product is. Those products are delineated and completely separated on there. Believe it or not, something is, when I first heard this from my quality control, I was like, are you serious? This is ridiculous. Vial container closure integrity tests. I said, what the hell is that? You got to send this thing out to make sure that absolutely nothing leaks in there. There's no bacteria in the actual container that you're actually using before you even put a product in there, in that specific container. It's costly. These tests are really, really costly. The certificate of analysis, who produced certificate analysis, ask for these things. You know, even if you're producing your own product, if it's autologous or whatnot, produce a certificate analysis and give this to your patient. It shows honesty of what you're doing. It shows that your work is being done the right way, and you're doing this. And any supporting documents of such, too, of what you're going to be doing. You should be doing this. Validating and testing. When we talk about off-the-shelf products, and we talk about production of products, period, it's up to you, and every single product has a standard or a specific standard that you set up, meaning the individual that's making the product. You set up the product so that you validate this and you test this. You produce different standards, which we'll discuss in a little bit, of particular, like some of these off-the-shelf products that you'll see, and that you're maintaining the program, meaning that you're checking this standard on a regular basis. Let's say for every once a month, you go back and you check that the quality is there and the standard that you produce is there. In some cases, is the product red? You know, when you look at red, when anybody sends me a red product or anything, I test a lot of these different products. The first thing I look at is, I ask a question, is the product frozen? Does that product have blood in it? And the second question is, does the product have DMEM in it? DMEM is a media that we use actually to cultivate cells or to make cells happy. But the problem with that is that it's actually a research-based product, that's one. And secondarily, is that it mimics, DMEM mimics the estrogen receptor, so it could be, I mean, you got to watch it if you're using this kind of stuff. Was the product independently tested? Who did it, and was it done correctly? Read this stuff well and look for discrepancies like what we just talked about a little while ago. Company comes to you and sends you a booklet of different factors, and you're like, oh, wow, these guys tested 200 different things. I really don't care. You test it. Show me what these 200 different things do. And why did you test these 200 different things? Why are these important to have in the product? That's what's important. State standards, making sure that you're maintaining or the product is maintaining state standards, meaning pathogen serological testing is done. It should be produced in a GMP manner. Every one of these products should be produced in a GMP manner, and that program, like what we just discussed about maintaining that clean room, is maintained. Meaning you're maintaining the validation, you're maintaining that clean room according to standards. Testing. So when we discuss testing and people talk about viability of cells, cell counts, how was it done? This is important because in the old days, and I still do it every once in a while because I have fun doing it, is I use this thing called the hemocytometer. Does everybody know what a hemocytometer is, or a Neubauer chamber? You guys are familiar with this thing? It's a little tin, a little glass thing, and you use this stain called trypan blue. It's a blue stain. And you look underneath the scope, you put, let's say, like 10 microliters of sample and 10 microliters of the stain, and underneath the scope, you will see a grid. And in the grid, you'll see all these dots of cells or debris or whatnot be in there. You have to be able to identify the cells, and you have to be able to quantify it. So then you count these grids, these squares, count, 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 and you'll be there with the count. It's a very, very subjective count. And if you don't know how to identify a nucleated cell, if the product has red blood cells in it, you will be counting a lot of red blood cells. The viability will be much higher than it actually is for the product, and you'll have a lot more cells than you actually think. This is really, really common. When I first started hearing stromal vascular fraction, people talking to me about billions of cells, and I was just like, this is impossible. So I had to get in and dig in and study this stuff, and it's because they use these type of systems. You want to look at cell counters similar to this, or a flow cytometry, where you quantify, like for instance, this is done through what's called acronin orange and propidium iodide, where you quantify the nucleated cells that are in there. This type of cell counter basically looks at alive cells. The nucleus is live, the nucleus is fine, and then it quantifies the outside if the cell is dead or not, the outside of the cell. And it tells you, a green image is these are all the live cells, and if something comes out and down here, which is a reddish, it tells you what is dead. You can't really see this, but there's some red dots here. You can't see it that well. You know, for instance, in this particular one, the product is 66.1% viable. These things give you the size of the products. You know, live were 7.4, total cells are 6.9, and the dead cells were 5.5. Remember, dead cells are blebbing. They're starting to bleb. This tells you that any cell that's removed or eliminated is gone. The nice thing about this type of counter in the product is you can see cells. You don't see a bunch of debris in there. And that's actually the really good thing about this. Flow cytometry, you want to identify specific cell types, and you want to have an idea what's in your product. And that's the concept behind this. What is in there? And this also helps you confirm what's live, because to do this procedure requires you manipulating the cells that are there, requires you to add specific agents in there, and you put the cells in a harsh environment to do this. So actually on these type of tests, the viability actually goes down a little bit more on a product, because you've now centrifuged the cell. You've now added different agents in there that you want to tag with an antibody to run. What type of markers do you want to look at? For instance, this is a cord blood product, and we want to look at the amount of immune cells in there. What we discussed earlier, CD45 being a gross overall immune cell type marker, CD34 and CD45 positive. This in combination means this is a hemopoietic stem cell, is 34, but you don't run this by yourself because this marks other cells too. You mark it with this, and to make sure that's of the immune origin, there's only 0.5% of them. That's still a lot. It's quite a bit, because the population of cells that you're in there is quite a bit. CD73 is maybe one of the markers that we discussed, CD90 of mesenchymal stem cells. And this is only done here with these two markers of MSCs. And this suggests, listen to that word, suggests that there's mesenchymal stem cells in there. The only way I go back and prove it is if I take that population, and what does it have to do? What do we talk about? It has to stick to plastic. It has to do that. Okay? This just suggests based on those markers. But remember, this is a heterogeneous population of cells. I don't really care. I know the product works. HLA-DR, which is basically remember, it's an MHC class 2 molecule. There's 17% of it in this particular product. HLA-ABC, there's 47% of it. This is what's considered somewhat, 17% is negligible, and it could also be, sometimes these flow cytometry, you can get cross-reactivity. If a cell's undergoing death, it can label non-specifically, and you may have sometimes these type of problems. In this, your questions are going to be, what is the mode of repair? This is why you want to look at specific things in a particular product. This product, we looked at 4 very important things. And we looked at, remember the strong mitogen, which we've talked, and I've been hopefully drilling home with you several times, FGF basic, which is also called FGF2 if you hear that term. And it had it in picograms amount of 131 plus or minus 35. IRAP, which is one of the strongest anti-inflammatories in your body that suppresses that strong pro-inflammatory TNF alpha response, has a lot of it, 25,000 picograms, which basically boils down to 25, that's 10 to the 15, which boils down to 25 nanograms per ml. Stem cell factor, which is what activates your own endogenous stem cells, and vascular endothelial growth factor, 796 picograms per ml. Here's this stuff charted, so you understand that these have a mode of action, and these are what you're interested in seeing in a product. That's why we're not targeting looking at 30 different things or 75 different things. Because if you're going to show me the good, show me the bad. I'd rather you just show me the good and that's it. Let's be positive, right? Documentation. It's important that you ask for as much of this as possible. That you go through and whatever you're doing or whatever, if you're producing your own product, if you're making this, produce the correct documentation. Have that informed consent. Have that certificate analysis. Have the documentation that protects you and protects the patient. Both things. It's about both people being protected. When the product is being produced, make sure they have a system, what I call more than anything else is, well, part of the quality system, even including, believe it or not, a simple packaging labeling system that enumerates what comes from what. Material system, this is very, very important. Anything that we buy, believe it or not, as scientists, our regulatory people, we call them police officers, and sometimes when we see them coming around the corner, we run the other way because they're so hard to deal with. But that's okay, they keep us in check, and that's what they're supposed to do. The materials that we take in have to be from a qualified vendor, meaning that before we can even order it, we can find whatever we want on the internet, or we can find whatever we want with even Fisher Scientific or whatever it is, big companies. We have to make sure that that company has a quality system in place. Our regulatory people contact that company. So sometimes we want something overnight, impossible. Our regulatory people have to contact their regulatory people, and they have to demonstrate that they have a quality system in place. If there's no quality system in place for them and they not have validated their products, we cannot purchase that product, we cannot use that product. We've had products that we have some scientists order on their own, and it just sits there and it's in quarantine, because the one that releases the product is the regulatory people, the quality control, the quality assurance individuals. Laboratory controls in place, facilities and equipment system completely calibrated and in place. Most importantly, when you're using whatever you decide to use, pay a visit, ask to pay a visit. If somebody tells you you can't pay a visit, it's a problem. Something's being hidden, something's being done, something is not correct. Request that documentation and overview, make sure that you ask for copies if you need them. Ask the question, who produces certificate analysis? Is this done in-house? If you got data from somewhere else, is that added into your certificate analysis? Is this product safe? What are the standards for the particular product? How did you test the product to make sure that it's actually safe, or you actually showed that this product secretes whatever it may be for the product that you actually use? So in summary of what we just discussed, these systems are designed to protect you and designed to protect the consumer. Don't think a lot of times that the FDA, they overreact or something, but first and foremost is the most important aspect is safety. People discuss with me like, I want to do this, I want to do that, and I want to do that. I said, hey, you know what? The first thing you got to do is slow down and take measures, and you start off with baby steps of what is safe and what has been shown to be safe before you go on to the next step of trying something else on that actual patient. So it's actually first. Ask questions and be comfortable with who you're using, who you're dealing with, the team, etc., before you decide to take that leap. Please don't look at the money, because money comes with time and money comes with hard work. And obtain all the documentation that's necessary to support safety for your own protection. This is the most important aspects of it. Thank you very much, and ask all the questions you want. I'm here to help. Yes.